All right, good morning. We will get started with this. Before I get going, any questions? Am I going to give more details about the extra credit paper today? Uh, yes, let me close this down. All right, good morning. We will get started with this. Before we get going, any questions? Hang on here. Am I going to? Technical difficulties. All right, come on. Too much. Okay, I think I'm back. I think it's not going to talk over me. All right, so yes, there are some details I want to provide. I was trying to flip back. I need to stop sharing. Um, Back to my Moodle page. Let me, where is this? Too many buttons, I apologize. Okay, I'm going to share this. So the extra credit paper, this should uh, open up. So it's due on the 18th at 5 p.m. This is under the 15th through the 21st, that week's tab. I put it on there because that's when it's due. So you'll need to scroll through or scroll down a little bit to find it. Once you click on it, I think this maybe will get it. We will see. Well, the figure should pop up. It's probably trying to download all of this. So it's a figure that we showed before with the three different things looking at Medicare versus or Medicaid versus commercial insurance and perceptions with that. So a couple of things to think about. If there were universal health care, everyone would have the same level of care. Look back to that figure and address each of those points and think a little bit more about your own idea. Would you utilize more or less? Would you expect more or less out of it? And what would your attitude about well, it was all antibiotics in the first place. Your attitude about antibiotic use be different if the situation were potentially different. So a couple 1 to 2 pages, like I said in lecture, there's no right or wrong answer. I don't, everybody's got their own opinions with this, and that's all fine and good. I'm just kind of curious, as I looked at that, what would it be like? And seemed like a pretty simple way to get some points. When I was looking at this, my wife spouted off, what if it was universal, but you had to pay something? What if going to the doctor, it cost you $20 or something like that? Not a lot, but a little. Would that make a difference? And I'm like, well, I'm already done making this assignment, but I like the thought of that as well. Would that alter it? I'm not throwing that in there. I mean, you can think about that as a sidebar. Maybe that would make it a little different. Maybe not. Okay. Let me stop this one. Go back to this. Actually, let me share this one real quick because I wanted to share this. So we're starting chapter 17. I've got everything all out of whack here. So I'm sure at home you are seeing the table out of the chapter. I assume if not, somebody pipe up and tell me. It looks like this is what we're sharing. It's in green. So chapter 17, we want to think about really starting to think about immunity, thinking about our potential to mount a response, or as we progress through, maybe the lack thereof. So initially, I was a little bit upset because I had opened this chapter up a couple of days ago. I was going through, I was making some modifications. I was putting a few tables into the PowerPoint because I feel like they ought to be in there. I had almost all of this stuff done and I clicked save. Maybe it's because I was dealing with this at my house. I don't know. But I've dealt with it before and it's worked fine. Something happened and the file did not save as I thought it would. I opened everything back up and nothing was there, just little question marks. That doesn't make sense. So that's fine. I'm shooting from the hip. We're going to just use the textbook, and I'll provide some of this as we move through. So we'll bounce back and forth a little bit with some of the sharing because I feel like a few figures in there were a little bit blurry, and I was trying to get those swapped out. Maybe that's where it all crashed on me. So as far as the non-specific or the innate defenses, there's a handful of different things that we think about. These are our body's first approach at dealing with potential foreign invaders. We've got all sorts of capabilities of trying to protect ourselves, trying to prevent these organisms from causing issues in the first place. So we'll go through and hit on some details with the specifics in this table in this chapter. And then we will move into the next couple of chapters thinking about more details with the actual immune response, where we think immunoglobulins, things like that, that serve more of a protection. So initially, we can break down the innate into really three different main categories: the physical, chemical, and cellular defenses. And there's a handful of potentials with each of these that, again, we're going to touch base on as we're progressing along. So initially, we think about stuff like physical barriers, the unbroken skin, providing a great barrier to the outside world. And when we see issues, wounds, somehow opening that barrier for the world to come in, we see all sorts of potentials for organisms to break through that barrier and to truly take hold. Organisms that may be on the surface of our skin are an advantage and are part of the normal flora, helping to keep us healthy. If they get the opportunity to move through that barrier, they can be a serious pathogen. Thinking, like Staphylococcus aureus is a good example. So I'm not going to spout off all of these things because, again, the PowerPoints all deal with it. But there's mechanical defenses, your own microbiome, as I was just kind of hitting on with your normal flora, all providing some of these initial physical lines of defense to prevent some of these foreign invaders from actually taking hold. We move from that point to some chemical defenses, some of which might be really part of the microbiome, providing some potential advantages, and then our own body providing some other types of chemical defenses as well. So chemicals and enzymes within secretions, antimicrobial peptides, not antibodies, but small fragments or partial proteins, if you will, peptides, not quite big enough to be a full protein. So these substances have definitely some potentials for being bactericidal, killing off some microbes. Cytokines, we'll get into a little bit of detail about those guys, some chemical messengers that allow for cellular communication, kind of telling other neighboring cells there's an issue going on. Inflammation is kind of a double-edged sword. There's good and bad with that. And definitely when we think about potentials for these cells, these foreign invaders to be causing issues, inflammation helps this first kind of arm of the response to say there's issues, other cytokines are secreted as well to tell other cells there's issues, and everybody starts kind of gearing up to defend. And it's pretty impressive with all of the potentials that go on with this. And there's also circumstances where maybe you become overwhelmed, where the defenses, the messages being sent out are too extreme, and that causes things to kind of go a little haywire. When we get into the cellular defenses, now we start thinking about stuff we'll describe a little bit later as some cell-mediated types of immunity. Cellular defense makes sense. There are numerous types of leukocytes, white blood cells, the granulocytes and agranulocytes. We'll go through some details dividing these guys out and talking about some of their specific properties. And they play an important role in defending ourselves. And as we move into later chapters, the cellular defenses will be described in more detail because they do play an essential role. Let's see. I know in the PowerPoint, this is the very next slide, since I'm already on this in the textbook. I'll just leave it right here, and we'll talk about this, and then I'll shift gears over a little bit. So this should be some review as far as like biology stuff goes. This is something that's discussed there. I feel like you get you know, a bit of redundancy as you move through different biology classes where you see things kind of over and over. But that ought to raise a flag and say, you know, there's some importance to this. When we think about the ways, and we're thinking our own body, the ways that multicellular organisms, the way the cells are linked together potentially lends to all sorts of different types of interactions. And when we think about different organs or organ systems and the types of cellular linkages, if you will, are varied. You're not going to see all three of these, maybe, but typically not all three in one type of cell. You're going to see this kind of being varied. So initially, the tight junctions, this first bit right here, we've got all of these little blue dots. It says in here in the text of the figure, these are kind of like little rivets or maybe spot welds. I like to kind of think of it more like caulking around the tub or the shower to keep the water from getting behind the wall. That's what you truly want in your shower. You don't want that water leaking back there. So this provides a tight seal, as tight junction implies, something that helps to keep materials intact. Intestinal cells are a great example of cells that have tight junctions. You want the food stuff within the lumen, within the inside of the intestinal cells, and you don't want it leaking out of those cells into the abdominal cavity. That'd be bad news, all right? A stinky situation, all right? You don't want that going on, all right? So the tight junctions provide this barrier to truly kind of seal these cells together, all right? The desmosomes, the guys next door, this is kind of more like Velcro, I think about. I think the figure mentions like shoelaces. Loose, but it still holds stuff together, but loosely. So the potential for things to move between these cells or through is considerably greater than if you flip back to the tight junctions, all right? So this kind of more to anchor, to tether, to hold cells in place, again, loose. The gap junctions, these are little holes, little portals that allow for molecules to move through these cells to allow for communication between the cells. And so again, different types of situation where you would want that that potential to occur. And so all sorts of different capabilities that these cells have. It just depends on the specific situation. All right. I'm going to switch back to the PowerPoint. Stop that one. Get back to this. Okay. So make it full screen. So there are all sorts of, again, things we want to think about as we progress through this chapter. Initially, chickenpox is the first figure in the text that there's a little tidbit about it. But varicella zoster is the virus. It is very contagious. Coming in contact with someone, if you have not had chickenpox or the vaccine, coming in contact with someone, you're almost guaranteed that you're going to contract it. The amount of this now is, I think, considerably lower with all of the vaccination efforts. We don't see huge issues with chickenpox. It's relatively was relatively common childhood illness. You pretty much just knew this is something you're going to get. And now with vaccines, it's, well, hopefully something you assume you're not going to get. So there's all sorts of little pustules, pox-like lesions, and it's really an inflammatory response, part of that innate immunity that gets initiated. And there's all sorts of potentials that start happening with this immune response, and the recognition of virally infected cells stimulates part of this immune response to seek and, well, seek and destroy cells that they recognize as being plagued by this virus, or any virus for that matter. There are mechanisms that try to deal with that. Obviously, not 100%, right? So we already saw that, the desmosomes and gap junctions and tight junctions. So this next figure is a cross-section of our skin. As we said, with the initial first lines of defense, we think of the unbroken skin as a great barrier to the world around us. So there's 3 layers, and you don't need to truly get bogged down with all of the anatomy of the skin, just to recognize that it is the unbroken skin is a great barrier for that. So the epidermis, the outer layer, the dermis right under that, and then down below the hypodermis. So 3 layers thick. The epidermis, numerous layers of these dead skin cells to the top, which provide this barrier, if you will. As the epidermis is, continually kind of being sloughed off, the microbes are also kind of removed with that. The surface of the skin is a pretty good habitat for a lot of different types of microbes. And we can also kind of think of some micro niches, some areas of the body that are more, more hospitable to some organisms than others. You don't see the exact same profile of microbes on every single square inch of your skin, right? There is definitely some variation with that. And you also need to think a little bit more about the microbe's ability to tolerate that relatively adverse environment, right? The skin is kind of like a desert, right? And there are definitely some potentials for things to thrive in those kinds of conditions, and then maybe not so much. Okay, so this next image, this is kind of a little bit gross. The gentleman's arm is, it has some serious lesions. Sporothrix shankii is a type of fungal organism. It's one that affects the skin, as we're seeing right here. There are a handful of different types of fungal infections, the dermatophytes targeting skin, and then we can move in and think of other, maybe systemic types that can move throughout the body. Sporothrix is one that targets the skin. And it's got a common name of rose gardener's disease. It doesn't have to just be roses, but it is a relatively common fungus that you see on colonizing on these plants. And so getting scratches or scrapes potentially inoculates that into you, and then it grows. Yeah.
Do you happen to know if those types of funguses that live on the roses, do they live on all roses? Is it like really mutualistic relationship that they have with roses?
That is a good question. So the question was, does this fungus that lives on the rose, is it a mutualistic type of fungus? Are they benefiting the rose or not? That's a good question, and I am not certain if Sporothrix growing on the rose provides a benefit to the rose. It's a good question. It kind of makes you wonder, maybe there is some sort of benefit that that fungus is receiving from the rose. I do not think that the fungus causes ill effects on the rose plant. So that would really kind of beg the question that maybe there is some sort of mutualism going on. But I'm not 100%. Us, when we come in contact with it, that's definitely not a mutualistic relationship, and we do see some serious pathologies with this. It is something that we can see in Idaho. There are, there's a handful of, when we talk about bacterial stuff, well, there's all sorts of potentials with that. We start thinking about other stuff, lots of different types of parasites. Well, maybe we're in a pretty good situation here where it's not very common to see that. Rose gardener's disease is something that we could see around here. This fungus does survive our relatively harsh winters, not that we had one this year. So it is not just found in roses. People have contracted this as they're pruning like shrubs, junipers and evergreens, things like that, that you potentially brush up against, and it scratches you a little bit, breaking that barrier. If that fungus is on the plant and it does scratch the skin, it inoculates it in, and then this fungus kind of thrives. It does take, to see this occurring, this is something that has manifested over the course of weeks to truly present like this. It's not something that just shows up right after you've pruned the roses, all right? So definitely kind of a nasty looking infection. So another potential mechanism, as we think the mechanical types of means for providing some mechanisms for defense, we could think about ciliated cells, like within the trachea, that help with a flushing action to push the mucus away, and really the potential for microbes that maybe were within that, you get them up and out of there. So this, again, a mechanical means where we've got this movement that helps kind of flush away those possible foreign invaders. Here, these goblet cells are cells that would be responsible for the production of mucus in the first place. That's another mechanism that we think about. with some potential to help provide protection against some of these foreign invaders. There's also, um, we're not going to get to it right at this bit, but within the mucus secretions, we do see some immunoglobulins as well, types of antibodies that are found within these secretions, and that also provides some protection. That's different than this innate response that we're thinking about right now, right? But it is part of the overall response. And, as I'm saying that right now, I want to stop and just mention this real quickly. And, in immunology, in the actual immunology course, we talk about these things as kind of separate entities, separate things going on. These chapters in the microbiology text are separate, talking about them as individual things. When we think about an immune response, we don't think about it separate. All of these things act together to try to give us an overall approach for protection. It's not just, oh, we'll task this one thing, and if it doesn't work, well, we'll call out this other thing. We call them all at the same time, typically, all right? There is a little bit of variation with that, but as we're talking about these things as separate sorts of ideas, you want to think about it, the big picture. They all are trying to work in concert to ensure the success, our survival. All right, that's the idea. All right, so these goblet cells are basically mucus secreting factories that have the potential to provide another barrier. Other secretions, tears help to flush away microbes within the eye. There are substances within the secretions that also have antimicrobial properties. We'll hit a little of that in a bit. Urine flushing action with that is also a potential to help remove some of these organisms. That's not always 100% effective, as we do see, UTIs where organisms colonize. And we'll hit that stuff a little bit later on as we start talking specifics with the disease. Yeah, so far as I'm going through here, all of these figures look just fine. So that's all fine and good. Somewhere it'll be blurry. I know I saw blurry stuff in here. This other secretions, the sebaceous gland secreting sebum. This helps to lubricate and protect the skin. It's oily secretions from some of these invaders. It also serves, so we've got chemical mediators that help to prevent some of these foreign invaders, but it also serves as nutrients for some of our normal flora, some of the organisms we want to stay there. And these guys, as they're consuming the sebum, they produce oleic acid, and that is another mediator that helps to prevent some of these foreign invaders from taking hold. Exogenously produced, so it's produced outside of our body. It's produced by the bacteria. As they degrade the sebum, it produces the oleic acid. So that could be a great example of a mutualistic effect, where some of these normal flora being fed the sebum produce oleic acid to help protect us. And deep down, it's protecting them too, because it's keeping their real estate and their food supply to themselves. And that's maybe a win-win situation. So this next figure is complex, I would say, to really to say the least. Complement is something that we can spout off a few things here, and I think it's all fine and good, but to really think about it in detail is kind of beyond the scope of what we need to think about here. There is a tremendous amount of stuff going on, and there are more details still being discovered as we think about this. There are handfuls of proteins circulating in our body that belong to classes of proteins that we call the complement system. Uh, Numerous proteins, and they act in a couple of different ways. Really, the end result is to provide protection, trying to either target a cell or tag a cell to be removed, or to punch a bunch of holes in it and cause its destruction, its lysis. Uh, there's a few possibilities that the activation of these proteins can cause. There's also a couple of ways that the complement proteins can be stimulated. So there's, you know, it's more than just one mechanism that may initiate this. That's kind of it's a fail-safe to ensure that maybe, maybe one way is not working, we can still recognize it and activate it through another mechanism, right? And that becomes super important, again, when we're thinking primary lines of defense, right? They also play roles in other defense mechanisms when we start thinking about antibody-mediated responses or the humoral response. That can come in to this classical pathway, the antigen-antibody complex that stimulates these proteins, right? So a couple of potentials. Recognizing some substances from some microbes, we can link up, we can bypass the littlest bit, so we don't need an antigen-antibody complex. We can come in at this mannose binding pathway or protein and initiate the pathway at C4 protein. The alternate pathway recognizes some other types of cells as well, again, foreign situations, and we can activate the alternate pathway, again, not needing antibody to initiate it. All of these proteins, basically, we've got a whole list of these things, and the way that stuff is happening, they are listed C1 through C whatever. We're going through C9 up here. Like I said, there's more than just this, right? But this main bit, they're numbered based off of how they were discovered, and the way that some of this stuff is functioning is a little bit different, right? As you see here, we go C1 to C2 and then to C4, C4 to C3. Why? Because 4 was discovered after 3, right? And deep down, I'm like, come on, can't we just number them in the order that they're occurring and just say this is fine? Would we care? The person that discovered 4, maybe he cares, right? I don't know, all right? Doesn't matter, all right? It just lends a little confusion. When we think about this, it's ultimately lots of proteins interacting together, kind of like a spontaneous reaction where the product of one reaction catalyzes the formation of the next reaction, sort of, again, a spontaneous type of reaction. So doesn't matter how we're starting it, right? Handful of ways to start it. We get into this bit here, the C3 protein gets cleaved. It gets split in half to C3b and C3a. These proteins interact with the next player, C5. It gets split apart, right? As these proteins interact, the C5b starts forming, and this would be on a foreign cell, on a bacterium. The C5b initiates these other proteins to target and attack that cell. There is, again, kind of a chain reaction. All of these proteins come together and they basically bore or drill a hole into that foreign invader, all right? It causes little pores, all right? Porosity. And now that we've drilled a bunch of holes in it, that cell, the contents of it, leak out. It causes lysis. the membrane attack complex basically perforates those cells. So there's that. There's the potential for opsonization, where we will coat and put like a little flag, if you will, that says, this is a cell that needs removal, targeting, or stimulating maybe a macrophage to come in and destroy. That has other capabilities where maybe that stimulates that macrophage to present specific fragments of that cell and stimulates an antibody producing cell to start pumping that out. That causes this part of the cascade to be stimulated, and the cycle just keeps going, right? And that may have all been initiated, first off, with this lectin pathway before we ever produced antibody to begin with, all right? It really just depends on the circumstances, right? A lot of the time, we are thinking classical pathway where there's already been antibody. This antibody circulating in your body stimulates the protein complements that are circulating, again, within your bloodstream, and it causes them to become activated when they come in contact with the specific foreign invader. It's a lot of stuff, all right? You want to think, a couple of pathways, but the main idea really to attack, causing lysis or potentially opsonizing, coding, something that allows these cells to basically be recognized, right? They can also link up and target toxins and things like that cells might be releasing, but that's fine. So we mentioned the cytokines in that first bit in that table. These chemical messengers, there's all sorts of ways that cells may communicate. And there's huge amounts of cytokines, right? And what they're doing, who they're talking with, there's definitely some differences with that. So as far as these messengers go, we can break it into 3 bits, and really it's what, or not what, but whom they're speaking with. The autocrines, these cytokines communicate with the cell that releases them. Auto, the same, all right? So autokines or the autocrines produce cytokines that signal or stimulate itself, right? The paracrines, these guys are secreted to like close neighbors, right? Nearby cells, right? And so this secreting cell releases the cytokines, and you've got a nearby cell, different type of cell, but a nearby cell that has receptors that will link up to those cytokines, and it's like a switch. The cytokine links up to the receptor and it turns on or off something inside of that cell saying, holy cow, there's bad guys, make this, right? Or shut this stuff down, there's bad guys. One way or another, the cytokine says there's bad guys, let's do something about it, all right? That's really what we're thinking about, all right? As these guys are trying to stimulate whoever, all right? The endocrines, these things are released and they make their way into the circulation and travel throughout the body to distant cells. So farther away, but same sort of an idea with those receptors allowing something to, again, be stimulated one way or another, turned on, turned off, producing or inhibiting the production of specific things. And it is definitely varied, dependent upon the respective cytokines. All sorts of different triggers, if you will, to cause things to either, again, be turned on or turned off, one way or the other. So numerous potentials for cytokines and the possibility for their respective stimulation of cells, however they do it. So interferons are specialized types of cytokines, and there's a handful of different types of interferons. These interferons are messages that the cell is secreting that basically is a stimulus to others that, and there's varied cells that they may be stimulating, but to others saying, there's a virus, right? There's this type of a bad guy is what we're dealing with, and that's the message that the interferon is saying. All right, it's some sort of virus. So they may go this way and stimulate neighboring uninfected cells, and basically telling these cells, "I'm infected. Watch out." And the cell switches over, and any RNA will be destroyed inside of it. So it starts inhibiting protein synthesis, thinking viral, RNA coming in. We want to ward that off. We want to inhibit its synthesis before we become infected as well. All right. Uh, it may tell other cells, neighboring infected cells. All right, so here this cell is infected. The interferon is released. It links up to it, and it tells this cell, there's bad stuff happening. What you need to do, take one for the team. Apoptosis is programmed cell death. It's cell suicide. And these cells, knowing from the stimulus that they've received, that they're basically bad news, it tells them what you ought to do is just destroy yourself. That will prevent the massive production of virus to be released and infect other cells. So take one for the team and prevent that from being spread. Kind of a impressive strategy. There's also cytokines targeting stuff like macrophages, linking up and telling them similarly, we've got issues. The macrophage now may kick it into gear and start targeting these specific types of cells once it receives that signal, that stimuli. It's got that capability of responding and defending accordingly. Okay, let's look at this. This is one that wasn't so good. I can tell that just looking at it. I want to flip it to the book because I feel like it is a little bit better with that. Okay, so you should be seeing the figure. This is figure 1713 from the text page number. I can't tell you. I'm sure there's got to be a way to see what the page numbers are when it's on the online book, but I don't know. Matters not. So when we think about all of our cells that make up what we call the hematopoietic cells, we could say white and red blood cells, erythrocytes and leukocytes. There are numerous types of cells, but they all originate from this type of stem cell, a multipotent hematopoietic stem cell, a hemocytoblast. This is a cell that has the capability of differentiating into two different lines, and from that, further differentiating into whatever it's going to be when it grows up, right? That's the idea. So some cells stay as stem cells as this division's occurring, lending to the potential for them to keep proliferating this. There are definitely issues where things can go wrong, and we can see issues on either of these cell lines. So there is this initial differentiation that allows them to go down one path or the other, going to the myeloid or the lymphoid route. And once we start differentiating that way, there is still the potential for further differentiation. You are going down a path, but you've yet to truly choose exactly what role it is you're going to have. Still time, right? So specific chemical signals, cytokines, all sorts of different chemical messengers start acting upon these cells, giving it some potential thoughts, if you will, to differentiate this way or that way, chemical signals, all right, that allow it to proliferate or to differentiate one way or the other. So we'll look at the myeloid first because that's where I'm at with it. So with the myeloid route, we may go this way, this huge kind of amoeboid looking cell. This is what we call a megakaryocyte. The megakaryocyte will further kind of fragment. Really, it gets kind of destroyed basically in this process, and it becomes the platelets. Platelets look like kind of very small nucleated cells, but really they're fragments of this megakaryocyte. They're not a true cell. They're particles, all right? The erythrocytes, these are your red blood cells. The erythrocytes are the oxygen-carrying cells, and, well, carbon dioxide for that matter. They deal with gas exchange. And truly, when we think about all of the cells circulating around within the plasma, erythrocytes are the major players. And then we get the rest of these guys. Mast cells, kind of macrophage-like. They're not exactly the same, but you could see these within tissues. They become important a little bit later on. We'll see some of this stuff. They become important when we think about different types of allergic responses, inflammation, not just allergic responses, but inflammation as well. Mast cells have the capability of releasing histamine, which is a vasodilator, a key initiator of inflammatory response. The next ones over are the myeloblasts. The myeloblasts have the capability of being, again, cued by different chemical messengers to differentiate into a class of white cells, these leukocytes, the granulocytes that we see right here, basophils, neutrophils, and eosinophils. These all have different, so lysosomes have all sorts of digestive enzymes that help to break down stuff that's brought in, and in this case, maybe foreign invaders. There's different properties within the lysosomes in each of these three classes of cells, different substances that they harbor. And with that said, when we stain them up, they stain differently. The basophils are a basic staining. The granules are a deep blue, purplish color. The neutrophils, the granules, are neutral staining. They're kind of a lavender color. And the eosinophils are acidic staining, and they are kind of bright orange or pinkish colored granules, all the little speckles within there. And again, it's lysosomes. The monocytes, these guys, you don't see granules within them. The monocytes in the circulation appear like this, and then as they move out into the tissues, they differentiate, and you see macrophages or dendritic cells within the skin. And these guys seek and destroy, phagocytizing and destroying those foreign invaders. The granulocytes have some phagocytic capabilities, neutrophils especially. The basophils and eosinophils, maybe not so much. They do more extracellular killing. Instead of pulling in and phagocytizing, they will dump their digestive contents to the outside. Eosinophils are important when we think about maybe some types of parasitic infections. Basophils, we can see them kind of like a mast cell, and they're important in different types of allergic reactions and also some stimulators of inflammation as well. Let me flip it back up. The lymphoid line as we move down this way, so again, differentiating from that stem cell as we move to the lymphoid route, we've got a couple of different avenues that we may go with this as well. That lymphoid cell may become a natural killer cell, a large granular lymphocyte. It's got a lot of digestive enzymes within there, all right? And these guys are, as the name implies, natural killers. They are on a seek and destroy mission. They do not need antibody to stimulate this destruction, they can target stuff. And they look for things like maybe cancerous cells, potentially some virally infected cells might be targeted by natural killer cells as well. The smaller lymphocyte right here, this guy can differentiate into either the T or the B lymphocyte. Looking at them microscopically, although it looks like they're different here, looking at them microscopically, you can't tell the difference between these lymphocytes. You don't know, all right? They both are important in types of humoral and cell-mediated immunity. The T cell is really one, well, there's a class of T cells, but they serve to initiate different types of immune responses, telling cells like B cells, you ought to show up and start pumping out antibody, and telling some of these cell-mediated guys over here, telling some of these granulocytes the same sort of an idea. And like I said, oftentimes we see all of these things working together, all right? Some of these T cells help to initiate this response, but a lot of the time you see multiple players being called out to help. Okay, and I think that's good. We will talk about this table on Friday. It is something that I recommend you look at a little bit. There's details about all of these cells. I've already kind of hit that stuff, but we'll look at it the littlest bit next time. Okay, any questions or concerns? What am I going to post? Chapter summaries. I'm going to get those posted, hopefully, if not by today, by tomorrow for sure. You are welcome. Any other questions? Okay, guys, have a great day. See you Friday.